Thank you very much for your attendance. With that, I'd like to invite Maksim Plikus, who's one of our newest faculty members at the center and has really launched an incredible research program here. Welcome, Max.
Thank you. So it's really exciting to see that so late in the day, there's so many young people here instead of watching Netflix. You guys are here. This is really, really encouraging. So let's go back here. Okay, so let's talk about skin and scars. So are scars bad? Are scars bad? Depends. Oh, you guys are smart. Okay, yes, it truly depends. They're not really bad. They're really essential. Scarring is an essential mechanism of survival. This is a way we deal with wounds. And if you cannot close the wound, then this could be fatal. So forming scar tissue is fundamentally important. And it's a way to repair injuries. So also, scars sometimes can have really symbolic meaning, as shown here. So what you're looking at is a skin of an alligator on the left, and on the right is also what's called alligator skin. This is a type of scarring that is introduced purposely onto the skin in Papua New Guinea. But the mechanism behind it is quite fascinating because without really much science, people have figured out empirically how to achieve such a large protruding scale-like scars. And it starts with wounding. So you would take sharp glass, typically a rock, then you would introduce wounds. And then as you go from left to right at the top, so then you would apply mud that covers the wound, but it also infects it on purpose in a way, and then you repeat that. So you repeat infection with muddy water. So you splash your wounds with muddy water, which actually delays wound healing, infects the wounds, and make them fight more. And as you can see here, you would actually see quite an infected wound. So it takes a long time to heal, much longer than non-infected wound would otherwise heal. And the outcome is more excessive scarring that protrudes and give the appearance of scale that's actually highly desirable in that context. So you said it depends. So if scars are really large, then it could be a bad thing. For example, when people experience large body burns and they undergo massive scarring, what happens actually is those people lose a lot of sweat glands. So that the ability to thermoregulate through perspiration is now compromised. So people with large body burns that have lost a lot of sweat glands cannot really handle really hot weather well. So for many other reasons, including cosmetic reasons, if you are talking about open body surfaces such as your face, there is a strong desire to repair wounds without noticeable scarring. And what is a scar tissue? So let's take a look. So what you're looking at right now is an inverted skin of a mouse that was stained with bright orange dye to bring up adipocytes. This is fat cells. And black, dark are the hair follicles that are growing. So what you see all along the perimeter of the scar, which is in the center, this is normal skin. So you see that it's really packed with pigmented hair follicles, and there's a lot of fat. These are just two really important complex skin components that you can observe on this image. And none of this is present in a scar. All of this is gone. So scar really just lacks all this important complexity that develops in the skin during embryonic development. So the dogma was that this is the end result of wound healing in a mammal, scar, formation of this simplified substitute tissue. But people have dreamed about ability to regenerate lost skin completely. So here's an example of an Iron Man 3, okay? So here, superhero has 3 big gashes, and how did the superheroes heal? Sure enough, they set their arm on fire, it burns, and boom, it regenerates hairy skin, okay? So obviously this is fantasy, but how far is it away from reality? So what we know from studies done in embryonic time, for example, if you do an in utero surgery on a mouse and you introduce a wound during embryonic day 15, which is fairly early, and if you allow for mouse to be born, you will not be able to find that injury because it will repair scarlessly. It will grow haired and fatty skin. But if you wait just 3 days and you do the very same thing at day 18, which is about a day before mice normally will be born, you will form a very obvious scar tissue that will persist. So the concept has formed that the difference between day 15 and 18 is the plasticity of cells. Cells are much more plastic, they're more stem-like at the earlier time point, and as you proceed in embryonic development to a later event, you lose that plasticity. So now when massive injury is introduced, cells are on the injury side are not capable of regaining the plasticity that they had at earlier time. And regenerative therapies conceptually think that it might be possible to reverse the plasticity of cells at the later time point so that you take adult cells or more developed cells back into like early morphogenetic phase where you enable them to behave as if they're in day 15. So is it just a fantasy or is it really possible? Turns out that naturally adult skin, not even late embryonic skin, adult skin in many mammals, such as mice, display ability to behave like embryonic day 15 skin under certain circumstances. So if you were to introduce small wounds onto the mouse, they will stereotypically heal with the scar tissue that I showed you earlier. But as you start to increase wound, so you start to get meaner and meaner to those mice, but still within the approved regulations of the university, you make wound that's about 1 centimeter. And you allow it to heal completely naturally without trying to sew it, without trying to put bandages on it. It would actually undergo full partial regeneration in its center, not at the edges, in the center. So what you see here is a view of regenerated wound where you see a population of a couple hundred of hair follicles that are sprouting from the center of the wound. And they very clearly lack pigmentation, so they're gray hair, but by all other means, they're normal hair follicles. And you can see that right next to it, at the edges of the wound, there's lack of regeneration. So it's really restricted to the center. More interestingly, if you wait a little bit longer and you invert skin and you examine what's under those hairs, you will actually find that it's full of new adipocytes, the fat cells, which are the really important cell types in the skin. So what you see here, there's 2 lobules of bright orange adipocyte populations that regenerated. And the only reason they're there is because above them are those white hair follicles. And this behavior is lacking at the edges of the wound. So this is just magnified quadrant, upper quadrant of the previous wound for you to really appreciate how complete this regeneration is. So if you look at the regenerated piece of the skin in the lower right corner and compare it with the periphery where there's normal skin, you can agree that other than pigmentation of hair, which by the way we can correct for, it looks amazingly similar. And strikingly, separating normal skin and newly regenerated skin is this very simplistic looking scar tissue that lacks all of this complexity. So in my research, I've been quite fascinated by this natural phenomena, especially because it does not require any complex manipulations. In fact, you should not be manipulating wounds. You should enable them to heal completely naturally to achieve this kind of result. So I've been quite fascinated what's behind it, what's the mechanism. So as scientists, we really would like to know the mechanism, cellular and molecular aspects of this behavior. So, quite a number of years fast forward, we actually understand a little bit. So I will give you some bits and pieces of that mechanism. So we know that regeneration involves 2 consecutive events. First event is regeneration of new hair follicles. And those who know me know that I am pretty obsessed about hair follicles. So if you start asking me about hair follicles, you will probably not leave until about 11 p.m. tonight. So do not go there far. So they extremely complex stem cell rich mini organs that continue to regenerate in cycles over the lifespan of an animal or human. And the newly formed ones in the wound do exactly the same. They have very prominent population of stem cells. They regenerate in cycles. They do all the same things. And then the regeneration of hairs is followed by regeneration of fat. So we'll talk about fat a little bit more. So what do we understand about the mechanism of regeneration? One word, it's very complicated. I should say complex if it's one word. So morphologically, it looks exactly like what you would expect to see in embryonic day 15 skin. The formation of hair follicles, it looks like onion grown into the ground. So it starts off with a little seed, which is this small cluster of cells right here. And then give it a few days, it starts to grow down. It has this onion-like morphology, and give it enough number of days, it will sprout out new hairs. And if you don't shave a mouse, what you will see is just stuffed of white hairs covering the wound. You wouldn't even know there was a wound there. Other than pigmentation. And underneath the skin, this is the appearance of scar tissue. It's fairly simple. So it's covered by the structure called epidermis. This is the outer epithelial layer, right here. And underneath is fibroblast and ECM and blood vessel-rich tissue, scar tissue. But in the center of the wound, you have many more things. So you have those hair follicles. They extend deep. Those gaps that you see here, those are actually fat cells. And there are structures here which we call sebaceous glands. That's the only reason you have to shower, is because your hair gets oily. So these are oil-producing kind of satellite mini structures that associate with each hair follicle. So all of these things come back. So we know, and then about fat, which is the secondary event, it seemed to be highly 100% dependent on the hair follicles being there in the first place. So you need to regenerate hair, so as I was showing, and this process takes time. So this is in days after the initial wound was introduced. So fat cells, first fat cells only really appear by day 23, so you really have to be waiting. And they appear around those hair follicles, which for convenience are labeled here genetically blue. So these orange cells start to pop around the hair follicles, and they increase in number over the next couple of days. Only takes place around the hair follicle. So hair follicle is doing something. It's either providing progenitors for fat cells, or it's providing molecular signals. Mechanism of regeneration of the hair is complicated. At its core, it's really recapitulating embryonic development of hairs. So at its core, it depends on the very same signaling pathways. But what starts it is very different from embryogenesis. One of the really important cell types during repair of wounds are immune cells. And immune cells, such as regulatory T cells, which actually provide first molecular clues for the process, are not even mature during embryonic time, such as a day E15. So they're not even players during those early stages of embryonic development. But in wound they are. So the process of regeneration is not the exact replica of morphogenesis in embryonic time. It's partial replica, and it uses alternative mechanisms. So it's really inventive. So it uses a lot of immune cell assistance. And I will not go into too many details about this. Parts of this mechanism have been published, others have not yet. But once you have formed those hair follicles and they've matured, so now they look like this, so they're much longer, so they look like normal follicles at the edges, just lack that pigment. So hairs become a biofactory of all kinds of signaling molecules. So they start to secrete a lot of soluble growth factors, what we call it's a kind of a signaling molecule, into the scar. So in their vicinity, they enrich what other cells see quite significantly, and they enrich it for molecules that can induce formation of a scar cell, which we call myofibroblast. It's the version of a fibroblast, to acquire novel identity, to acquire an identity of an adipose progenitor, or you can call it adipose stem cell, which will then quickly go on to differentiate into a very large lipid-filled fat cell. So volume of a fat cell, so that you know relative to volume of a typical cell, it's about 300 times larger. So an adipocyte is about 300 times larger than a fibroblast. You can even see it with the naked eye if it's swimming in addition, it's stained. So down here is the piece of data. So this is a fairly complex genetic mouse model where we can label cells and we can later visualize them with the bright blue color, and we can track what they do. So it's a permanent labeling, and you can wait various duration of time, and you can see what are the blue cells becoming. So here we're labeling with the genetic driver specifically those myofibroblasts, and then we're waiting to see what can they become. Do they just remain myofibroblasts, or do they become something else? So shown here are new adipocytes that were teased out, microdissected, and put on a glass slide. So you can see that they have blue cytoplasm. So they were myofibroblasts before the time we labeled them. And now they have acquired a large lipid vacuole. So they are now adipocytes. So this is how we genetically track lineage and cell fate of cells. So we know that those new adipocytes in large wounds that I've been showing you actually derive from originally myofibroblast cells that are naturally not capable of becoming fat cells without assistance from hair follicle. What we also have recently learned is that not all myofibroblasts are the same. Just like in this audience, you guys have many different interests, and so are the fibroblasts. So in the last couple of years, one of the really revolutionary techniques has emerged as a major research tool. So everybody in this building is probably using it, called single cell RNA sequencing, where we can dissociate cells, live cells, from any tissue, and then we can sequence the transcriptome, so the mRNAs, at the deep enough level where using computer software tools, we can actually analyze cell types. And what is shown here is kind of like a typical single cell RNA sequencing experiment. So here we use skin, we dissociate live cells, we put it through this commercial platform called by 10x Genomics company, and then out comes the data. And once you analyze this data, you can project your cells and their similarities or differences onto this two-dimensional space, and you can color code clusters of cells. Okay, these are like gangs of cells, so to speak. So you got these blue cells, green cells, yellow cells, and the relative distance of cells to one another actually indicate their similarity. So if you see two cells that are right next to each other, that means they're probably the exact same cell type. But if they're really far away in this two-dimensional plot, they're very different. So analyzed here are just the myofibroblasts from the wound. And what you can tell is that we can detect with software analysis up to 12 subtypes. So really diverse. And we can further subcluster them by their identity. So we kind of know there's a logic to it. So there's this hierarchical tree that they form. So there's actually logic to all the subtypes. So one interesting finding that kind of came from this analysis, we found a subtype of myofibroblast representing approximately 13% of cells that had this really curious gene expression features that they look like blood cells called myeloid cells. So they had this hematopoietic signature, which is really confusing to us, but turns out that they are originally derived from circulating myeloid cells that migrated from circulation into the wound, acquired fibroblast morphology and gene expression signature. And we can visualize the cells, for example, in this another type of single cell assay called single cell Western blot, where you can analyze protein in individual cells. So what you see here is Western blot run from top to bottom, and magnified here are the individual wells capturing one cell. And you can analyze up to 2 to 3 proteins. So in this case, 2 proteins, they color-coded. So what we can identify is the reporter protein, red here, we call it M-cherry, and another protein, which is the marker for myeloid cell, called lysine. And what you can see is that cell in well 3 has both. And it would look like this. So you will have actually 2 spikes. You will have red and green spikes. So you would know this cell has this hybrid feature. It's partially myeloid and partially fibroblast. And when we track the cells using another genetic model, so now this is a little bit more familiar to you, so we label cells, we can later identify them as bright blue cells. So originally we labeled those myeloid cells, and then when we track them into regenerated wounds, and what you see here are these hair follicles and a lot of kind of large, like ball-like fat cells. So you see that a large number of these fat cells is now blue. It derived from myeloid cells. So that's what we found, that even remote sources can contribute progenitors for fat regeneration in wounds. So another really interesting aspect that we're interested, other than what are the cell lineages or what are the genealogies of cells, is who makes them what they are. So what are the signals? So I think in this case, regeneration of fat, it's very clear. Signal comes from the hair follicle. And we know that out of many signals that hair follicle produce, they produce the signal called bone morphogenetic protein, BMP. It's a type of signaling molecule that has multiple roles, but in the process of fat lineage formation, so when fat cells normally form from mesenchymal progenitors, it actually drives this event which we call lineage commitment, okay? It's kind of like in high school, your high school teacher could be the BMP, and if they truly inspire you to go into science and study hair follicle, then they will be the BMP, and this event would be commitment. So you will get committed to studying the hair follicle. And then you would activate something in your brain. In this case, these progenitors will activate this molecule, which we call transcription factor molecule. It's called zinc finger protein 423. So sometimes people come up with nice names. Sometimes people come up with really complicated names to the molecules. In this case, I think they just came up with a complicated one. So ZFP 423, although name is long, it's super important molecule because once it's been activated, it actually triggers the entire gene expression program that's different from myofibroblast. It's that of an adipose stem cell. And we can actually label those cells. So we know in embryonic development, all these blue dots here are those triggered ZFP 423 positive adipose progenitors that are actively forming and developing skin because that's when they form. In an adult skin, this process shuts off as it should. And only in wounds and only around those new hair follicles, which here look like this ghostly shadows, you see these little clouds of blue cells showing up. So these are these newly committing adipose progenitors from originally non-committed wound myofibroblast. And the hair follicle-derived signals, BMP signals, is the commitment mechanism. You also see here, for example, this vessel-like structure. So this is an independent expression of this molecule that's not related to myofibroblast, in case you're curious. And how do we know it for sure? Is this kind of evidence enough to claim what I just told you? It's certainly not enough. It's correlative, so you need solid evidence. So one way that we biologists generate solid evidence is by what we call loss of function or gain of function studies. You would need to mess up these things that I just mentioned to you, and you would need to see fat disappear or appear. Okay, that's how you know. So here's an example. We have a mutant mouse, for example, that has no ZFP 423 molecule. And they wounds regenerate lots of hairs. In this case, it's kind of convenient because with this oil dye, which we call oil red O, it just stains lipid indiscriminately. It also stains this oil glands in the hair, but they look very different. They lighter pinkish and they are larger. So each one of the structures is marking one hair follicle in the wound, but you see actually no fat cells. This is comparison of the control, not mutated mouse. So you see here those pinkish structures, these are the oil glands, and those small but really bright orange, intensely orange cells, these are the new fat cells. So here they have hairs and fat. So that's how we know. When we mess up that molecule, no fat can form, even though you have hair. So another example of this functional experiment. So in this case, people came up with a better protein name called noggin. So noggin is soluble antagonist for BMP. It essentially quenches it, so to speak. So we overexpress. So now in the mouse's skin, there's a ton of BMP antagonists. So they form lots of hair, and hair now is black, which is quite exciting. So we can rescue pigmentation. And there's only 2 adipocytes here. You can see right there, 2 tiny ones. So blocking BMP, not ZFP 423, can achieve the very same thing. It can block process of de novo commitment to fat. Okay, and this is again control. So spontaneously you can form pigmented hairs, but here adipocytes are non-compromised. Okay, so this is all mouse stuff. It's fancy and it's clean, it's genetic, but does it have any relevance to humans? So we wondered about this same question. So, and we performed some studies, in vitro studies. So one type of scar that you will actually hear probably more today in a second clinical talk are those scars called keloids. So there's a predisposition to this exuberant looking scars is genetic, but they are triggered by an injury. And once this injury is healed, scarring doesn't stop. It keeps on going. So it's almost like a benign tumor. It grows past the boundaries of the original wound. So stopping keloids is really complicated. So what we've done, we've collected primary myofibroblasts from keloids, and then we cultured them in what we call a adipogenic media. So it's the media that would drive differentiation of fat cells. So normally, you need to be already committed to fat lineage to be able to respond to this adipogenic media. One of the components is insulin, as an example. And only very few fibroblasts derived from human keloids can do that. But if you present them with human recombinant BMP4 protein, so it's one of the BMP ligands, that triggers massive commitment to adipogenesis, and all of a sudden you have many more orange cells in the dish. And we can also imitate this effect using hair follicles. So we could actually culture this keloid fibroblast across the filter that prevents cell migration but enables molecules to move in between. We culture them with micro-dissected human hair follicles. And where do you get that from? From actually, it's just from hair transplantation surgery. So people who get the hair transplanted, they're often quite generous. Surprisingly, they would donate 10, 20 hairs, no problem, to research. So we would collect those. And if you culture just for about 2 days hair follicles with those keloid cells and then induce them to differentiate into adipocyte, they do that readily. In this case, we use actually fluorescent lipid dye. So instead of orange, it's bright green and fluorescent. Same thing can be achieved. So this is quite hopeful because this tells us that although naturally fibroblasts from scars lack ability to form fat cells, by just giving them one molecule, you can re-enable this ability. So this could point toward new therapies for scarring. And another interesting aspect of fat, and wrapping up here, is so people typically now think, well, fat is not very good because many people have too much of it. They're struggling with maintaining weight, for example. But actually in skin, fat is really important and it plays many really important roles. It's also becoming prominently decreased with aging. So this is actually historic pictures of the same person taken probably a couple of decades apart. So you can see dramatic changes. So you can see aging effects, right? So what goes first? So hair is lost. Pigmented hair goes gray, but skin forms deep wrinkles. What's behind deep wrinkles? There's many aspects to it. So, but there is essentially the important aspect is deflation of the facial adipose tissue. There's multiple parts of adipose tissue. And this is an example of deep facial adipose tissue taken from plastic surgery book. I hope it's correct. There's major plastic surgeons in the audience. So I don't want to say things quite incorrectly. But this is a youthful face versus aged face. So you can see that those fat pads that actually make our skin look youthful are now really shrinking down because adipocytes have limited lifespan. They actually die off and new ones get restored from a progenitor pool that's actually limited. So given enough time, you would run out of your progenitors and the fat pad will now dwindle away like a deflated balloon. So then all of this excess skin volume will just deflate and you'll have these deep wrinkles. So perhaps one solution is to treat skin with one morphogenetic protein to induce fibroblasts that are still in the skin to become adipocyte and then blow up to about 300 times like a balloon to regain the volume. This is, again, still very theoretical. And last slide, why fat is really important. This would probably be quite unexpected for you, but it's been shown in a really prominent study that I collaborated on with an immunologist at UC San Diego. When you get an infection, skin infection, so bacteria penetrate underneath the epidermis, you have to deal with it. You have to fight it off. So typically you would think your professional immune cells will come to the rescue first, but turns out that first line of defense are the non-immune cells. And that includes adipocytes. So adipocytes have an ability to sense bacterial cell wall products, and they very quickly are triggered by them, and they start to secrete this molecule, which is called catalysidin. It's a type of an antimicrobial peptide. It's a molecule that would actually go to a bacteria and actively penetrate the cell wall of bacteria and form a hole in the bacterial cell wall, and it will just bleed the bacteria dead. So these catalysidins are produced by adipocytes that are triggered by presence of bacteria, and actually they provide a first line of defense. And if you have compromised adipose tissue in an animal models, the skin infections often lead to sepsis if this is broken. So with that, I would like to conclude and give a quick acknowledgement. So obviously work in a lab, especially mouse experiments, not only costly, but very time consuming and energy consuming. So you need very dedicated graduate students and postdocs. One of the person I would like to highlight is Christian, who is a star PhD student that started with me when I started here at UCI, and now he's a star postdoc. And he done major part of work that I'm showing you here, other lab members, collaborators, and funding. So I will stop here and give it away to our second speaker.
Next speaker is Dr. Victor Joe, who we're delighted to have with us tonight. Here, he's a burn surgeon and a trauma surgeon at UCI, and he's going to tell you about the clinical perspective in terms of wound healing and consequences. So thanks for joining us.
It truly is an honor to be here and to see so many bright young faces. You really are the future. Not only are your brains more plastic than mine, but you have the enthusiasm and you will be the future to continue the work that Dr. Plikus was talking about and some of the clinical work that I'm talking about. And so I hope that you are inspired through your schooling. It's interesting talking about the translation from basic science all the way to the end and what we do in treating patients. There's also a back translation, and it is not uncommon for me to point out when we're taking care of patients to go back and consider the physiology, the biochemistry of what we're dealing with. Because sometimes if you forget about that, we lose some of the understanding of why we do what we do and some of the decision that we make at the bedside for the patients. So I think having an understanding, at least a basic understanding of those basic sciences remain important. Why is that important? So as you go through school and wonder if this is going to be applicable, if this is going to be important, know that it's a building block to help you understand what you're going to be doing in the future. So again, I'm very thankful for the opportunity to come and to present. I have been a trauma, burn, and critical care surgeon for a few years. And my role currently is as the medical director for the UC Irvine Health Regional Burn Center. So, always have to start, I have no relevant financial disclosures. What I will say, though, are three disclosures. Firstly, you can't do a talk on burn surgery without certain photos that might be disturbing to those who aren't used to them, so that's just a little bit of a warning. Secondly, I will be talking about a couple of commercial products that are available to us that have come down the pipeline from regenerative medicine technologies. And then third, I don't know that I've ever been able to keep to a 20-minute timeline on a talk. And so I think I calculated that I have to do 30 seconds per slide, which I'm already behind. So with that, I think it's important to have a little bit of a background. When we talk about burn injuries, we usually talk about survival, and we talk about an LD50 or a lethal dose. And that is at what size of a burn injury will 50% of people survive, or conversely, 50% not survive. So if you look back in the 40s and the 50s and look at the different age groups, I just point out a couple. So in the very young, a 50% burn would result in 50% mortality. And it's the same for the age of 15 to 44, and as you start going up in age, even in middle age, it would only require a 27% total body surface area burn. Now, we only count second and third degree burn when we talk about total body surface area of burn, and I will discuss that again in the future. If you fast forward to now, these are data from the National Burn Repository, which is our national database, and you'll look just for comparison that in the 20 to 30 age group, that it would take nowadays a 70% burn to result in a 50% mortality. And in this 50 to 60 age group, it would require a 50 to 60% burn. We still have issues with patients as they get to the more extremes of age. The reasons for the improvement in the survival, of course, are things that have occurred in terms of advancements in the last 100 years. The one I would highlight, because it's relevant to us today, is early excision and grafting. Early excision and grafting became popularized really in the 80s after it was introduced as an idea in the 70s. And that was the idea is when you have a third degree burn that needs to be skin grafted, the advances in surgical techniques, in blood banking, in critical care, and all those things allowed us to start excising those wounds earlier and with larger surface areas with people surviving. In the past, what would happen is I wasn't there, they would wait for a process called suppuration. So that's when the dead skin, you start getting enough bacteria underneath, and it starts creating a separation from this pus-like material that would help lift it off. So of course, when we're waiting for that to happen, a lot of those patients would die of burn sepsis or widespread burn infection. So again, back to a little clinical background, you'll realize that most of us talk in terms of first, second, and third degree burns. What I want to highlight is that we like to think categorically, but it's really a continuum, right? So you can have a burn that is sort of on this deeper end of second degree, really acting like a third degree, and vice versa. So what we talk about are superficial, partial thickness, and full thickness burns. And to subdivide even more, we describe these partial thickness burns as either superficial, mid-dermal, or deep. And that's because these different depths of injury affect the time to healing. The time to healing affects the scar potential, and therefore, whether it goes into the first third of the dermis, the second third of the dermis, or deep into the dermis will affect treatment decisions. So that's why it's important for us to categorize them that way. And again, it's the depth into this second layer or the dermis of the skin. In reality, when we talk about needing to graft or skin graft a third degree burn, we actually do graft deep second degree burns. And that because by the time you excise the tissue to get to healthy tissue to put a skin graft, you essentially have no more of the important elements that Max was talking about to have timely healing. And therefore, to improve your cosmetic and functional outcome, if you no longer have enough healing potential to heal a wound within 2 to 3 weeks, the standard of care is to do a skin graft. So technically, deep second degree burns get grafted. Now, these are some examples. So this would be an example of a superficial partial thickness burn. This is probably superficial with some elements of mid within it. This is a deep dermal burn, and this is an obvious third degree or full thickness burn. So what has to happen nowadays is when you have a major third degree burn, that gets excised until you have healthy tissue. In this case, there's still some healthy looking dermis that is still present. So what is the issue? Well, the issue is in fact the tissue. So it could be, you like that, huh? It can be because of the size of the burn. So realize if we're going to take skin and auto-transplant it to cover that open area, you are going to be limited. The more you are burned, the less you have available to take. So the size of the burn, but also the depth of the burn. If you have residual dermis, that's a very good wound bed to graft. The deeper you go and then you start getting into, like this is a very deep burn, down to fat, now you're going to be putting a skin graft onto a bed of fat. And so we'll talk about why that's not necessarily the greatest. And then we're actually creating another wound in order to cover. So these donor sites now have to heal. That adds another physiologic insult onto the patient. And just like these can heal with scarring, now your donor site can also heal with scarring. So we compound the problem. The problem is that skin is very complex, and we don't think about it as being very complex until we realize just the sheer number of structures within the skin with a lot of different functions. So first of all, it's the largest organ in the body. If you are 5'6" and 140 pounds, roughly, you have almost 20 square feet of skin. It's an important barrier and a gatekeeper. It provides protection. Protection also with sensation. It helps us regulate our temperature. It helps with fluid balance, and it has endocrine function. So if you lose these components of skin, you lose these functions. And that, again, is going to create one of the problems we face in burn surgery. So the standard of care for covering these wounds is the split-thickness skin graft. In reality, there's many different types of grafts, but we're going to concentrate with 20 minutes on the split-thickness skin graft because by and large, that's a standard for treating third-degree burns of any significant size. So what we do is we take a dermatome and we shave off a split thickness. So we don't take the full thickness of skin so that the patient will still have enough dermis to heal that donor site wound. And then if we need more area covered, we will then mesh that skin, create like a fishnet and expand it. It could be expanded 2 to 1, 3 to 1, 4 to 1, even up to 6 or 9 to 1, depending on what we need to cover. And we can get fairly good results. So this is an example of a full hand, all 5 fingers and the whole dorsum of the hand. But in this case, I did not mesh. This was done as a sheet graft and tried to minimize the number of seams, because as you may be able to see, there's a residual scar from that seam from 2 different skin grafts being sewn on. And again, this is at 2 weeks. We don't know what, as time goes on, part of the biology of the healing of these grafts or healing of these wounds is the ability of the body to shut itself off. That's one of the issues with studying scarring, is sometimes the body doesn't know how to shut itself off, for lack of a better way to describe it, and we end up with what we call hypertrophic scars. And so the problem with scars is, this is an example, this was a little boy who was a victim of abuse, and so these wounds, they did not seek medical care, and so it healed with this very thick hypertrophic scar, if not a keloid. Other patients, we can do an excision and a graft, and you can tell there might be a little bit raised at the edges, but overall the contour is fairly good. This one, the contour, if you closed your eyes and put your hands across where it was grafted, you would not be able to feel a contour difference. But you can still tell that something happened, right? Because the pigment is not the same. What we want is this. And really, this is the right leg of this child. We want it to look like nothing ever happened. That is our goal. And that's a very daunting task. So scars are complex, both in their etiology, right? That whole last talk showed you how difficult it is to understand the full biology of how we normally heal, let alone what the mechanisms are for scarring, and then how we can then develop therapeutics to shut that down and to impact healing. Not only that, but in terms of what the patients see and feel, it is complex. So scarring is not just about a visual disfigurement in terms of a raised scar. It has issues with color, it has issues with pliability, and it has issues with the itch and the pain that come from a scarring. So it's multifactorial, not only in why they happen, but the problems that they generate. Not to also gloss over the fact that there are huge psychosocial and psychologic sequelae from having scars. It has become fairly understood from study of film, for example, that many times the villains or the bad guys in films have visible scarring. And this has become highlighted so much that even major publications have been studying, looking at film, and looking at the correlation for how beauty is depicted and how scarring is depicted in our culture. So healing burns and traumatic wounds without scar is considered the holy grail for us who do this type of surgery. Because we realize, for example, in one of those things where you talk about bedside manner, I've learned not to look at a patient post-operatively and look at it from my lens as a surgeon and say, that looks great. You say that a few times to a patient, especially on visible areas of their body, and they will tell you what they think about your great work. And so we then add to the psychological trauma because we're looking at it through our lens as physicians, that our work, that we have 100% graft take, and the surgery went beautifully. But if we realize that our goal is to have scarless healing, to have both form, function, and cosmesis, and all the functions we mentioned that the skin does, until we can restore every single one of those, our job is not done. And there's a lot more work to do. And that's the really big challenge that we face. And so that's why we look to regenerative medicine and the promise of stem cells to see if we can start understanding, because just transplanting skin has not been the answer. So how can we employ these technologies to try to get better outcomes? Well, I'm going to go over a couple. So one that we use is called cultured epidermal autografts. These have actually been around since the 1980s. And what we do is we take a a biopsy within the first couple of days of a patient with a major burn injury. So we have to take a couple of ellipses of skin about yay big, the full thickness, and we send it and they grow sheets of cells. However, remember we talked about the complex nature of the skin. They can only provide us sheets that are 4 to 8 cell layers thick. So it's only an epidermis, so it's an epidermal autograft. But those have been extremely important. So early on, it helped, for example, this patient with a 95% burn, meaning they only had 5% of their body that was not burned, and maybe even less of that to be able to take, right? Because if you have, for example, 1% right here, you're not taking skin graft from here, right? There's no way to shave with that dermatome. So just because you have 5% doesn't even mean you have 5% to use. So how are you supposed to cover someone with an autograft or their own skin graft when you have no skin to take? This was a review that was just recently published looking at 25 years experience with these cultured epidermal autografts. And it shows that when you compare, so the problem is they're comparing 2 databases. So that's already inherently some bias, but that's the best we could do for right now. They looked at a database where patients received the CEA, and they looked at the survival rates by percent burn, and they looked at the National Burn Repository, that same data I showed you in the beginning, to look at the survivability. And it's easy to see that with the use of CEA, especially as the burns got bigger, the survival improved because, as one of my colleagues nationally likes to say, burn survival is a race against time to close the wound. So with these big burns, if we cannot get them closed, patients eventually succumb. But you can see also the amount of take, it's considered successful if you can get anywhere like 70% take, which means you can take and place this on, and 30% of it will just not take and it will fail. And it's still a fairly successful surgery at this point with the CEAs. Okay? Have been other attempts to engineer a skin that is more robust and with more layers. So this is an example of engineered skin substitute. And this is by Dr. Stephen Boyce, who has been researching this for almost 4 decades and still been trying to engineer skin, again, taking a biopsy and then growing it over 3 to 4 weeks' time and having sheets of skin to be able to use. And he's been able to show in fairly small series of patients, and they mainly worked in the Shriners hospital system, but they were able to demonstrate fairly good cosmetic and functional outcomes using these engineered skin substitutes in comparison to a split thickness skin graft. The problem is, despite all this work, we still do not have FDA approval, and this is not commercially available for us to use. So I can't, for example, call his lab and say, hey, can I send you a biopsy of skin and can you make me 4,000 square centimeters of this graft so I can use on my patient. Other work that's currently being done right now by Professor John Greenwood in Australia, he envisions a 2-stage technique where he has developed a skin substitute that you put on immediately after you excise all the burned skin. It takes roughly anywhere from 2 to 4 weeks to mature, but during that time, via a skin biopsy and with a skin reactor, he's able to grow sheets of skin so that when this becomes mature, you then graft. And so what his vision is that by doing this 2-step approach, using the necessary time it takes for this initial skin substitute to fully engraft itself and take, that we could perhaps obviate any need for taking large sheets of skin to cover a patient. And I was recently just in Australia where his group presented some of the early work to demonstrate this proof of concept. So I will look forward to that research. The product initially to do the first stage is something that has become FDA approved for us to use currently, and we at UC Irvine are using this product, but what we're doing currently is still skin grafting with split thickness skin graft over it as our second stage. Now getting into more modern regenerative techniques, this is also a technology that was developed in Australia. It's been available in Australia and Europe for a long time, but only recently did the FDA approve it for the United States. Some of you may have seen a National Geographic talking about spray-on skin. That was something that was a little bit different, but the same idea. This is a process that Dr. Fiona Wood developed, where we take a small sample of split-thickness skin in the operating room, and on the back table in the operating room, we process those squares of skin, and there's an enzyme that breaks down the epidermis from the dermis. It breaks it down, and we scrape all that epidermis off, and then we filter it out, and it creates what they're calling a regenerative epidermal suspension that we then spray. And for each square that we take, it can cover an area 80 to 1. So we talk about that mesh expansion I told you about skin grafting, where I can go 2 to 1, 3 to 1, 4 to 1. This helps cover an area of 80 to 1. But it's only cells. It's not full thickness of skin. So how useful is this, and how are we currently employing it? Well, first of all, they were able to show that what you're getting is all the keratinocytes. Okay, the keratinocytes are what form that epidermis. But there are also fibroblasts and melanocytes, so it's unclear exactly what role these are playing in the overall healing of the burned skin. But this is one example of a patient who had a deep partial thickness burn to the face, where it was then surgically excised down to healthy tissue, sprayed on skin, and you can see the maturation up to 1 year and the cosmetic result. Secondly, we talked about widely meshing skin. If we widely meshed at 4, 6, or even 9 to 1, imagine again the amount of time it would take for all these interstices to fill by the natural healing process. It would take a very long time, if at all. And again, remember, the longer it takes, the more scarring we have. So another use of this is to go ahead and widely mesh, spray on the skin, and you can see it helps close these interstices quicker, therefore leading to better cosmetic and functional outcome. So we have begun using this. We have tried it on the deep partial thicknesses, like on the face, to see if it will heal. We're also using it in our patients with larger total body surface area burns to see if we can now take less skin or do less surgeries, mesh the grafts wider, spray on the skin, and still get an acceptable result. And so far, we've been pleased by what we've seen. We were also very fortunate to be part of a trial looking at something called StrataGraft. StrataGraft is a biologically active, full-thickness, allogeneic living skin substitute. So what happened was they discovered a keratinocyte line that was immortalized. So most of these cell lines of keratinocytes, they'll grow and divide for a certain amount of time, and then eventually that line will die off. But they discovered a line, and they called it NICS. Now, NICS does stand for something, but you see how clever they were. It's skin spelled backwards. But anyway, so they are able to grow this epidermal layer of keratinocytes, put it on a dermal equivalent, right? So just like the skin has 2 layers, this dermal equivalent has fibroblasts and collagen, because we need a carrier to take that. So the difference between those cultured epidermal autografts, that 4 to 8 cell layer, very fragile skin graft, now this is going to be a little bit more robust because it is on a dermal equivalent. So it could be stapled on, sutured, and placed. Now this garnered the attention of BARDA. BARDA is an offshoot. Whoa, that's like the hook.
That was a complete system failure.
Oh my goodness.
Let's see if we can regenerate it.
So BARDA is a branch of the federal government that's working in disaster preparedness. So in 2004, in response to the events of 9/11, President Bush signed the Project BioShield Act. And that generated monies to be able to fund research in order to treat patients who might become injured, for example, from a suitcase nuclear bomb. Because in those kind of cases, they're preparing for 2,000, 20,000, 200,000 injuries. So how are you going to do that? So they won up to $247 million of funding to help study the stratigraft. Sorry, we're going to have pictures. So the early studies were first an initial description published in 2000 of this technology, so almost 20 years ago. Then a phase 1 to 2 study comparing using this skin substitute as a skin substitute, comparing it to cadaver allograft, so donated cadaver skin. Because currently we're still, if we have too big of a surface area to cover with the patient's own skin, we have to cover it with something after we excise the dead tissue. And currently, many of us are still using cadaver. So that was the first step, was to compare its use with cadaver and would you have a good wound bed. In the process of that study, not only did they find that it provided a good wound bed, but they noted that some of the wounds did not need skin grafting. So that generated this thought, could this skin substitute actually be a single stage where it helps the body to regenerate skin? So this study, do I have to wait? Oh, so this study that we were able to participate in was a phase 3 study to try to get FDA approval, accumulating 70 patients with deep partial thickness burns who, by standard of care, we would excise and do skin grafting. So what we did was we paired and had the patients be their own control. So we compared two areas that were relatively the same. One area we would do a skin graft, in the other we would put this stratigraft. Okay. This was, again, some of the early studies where they demonstrated that it actually did lead to some healing that was similar to autografting. So this is the phase 3 study. 71 patients, 18 years older, anywhere from 3 to 49% burn. Again, patients were their own control. So we had completed this study, and they released for the public what they call the top-line results. So we haven't published the actual paper yet, but these are the public data that have been generated. So it was very successful. On average, 4% of the stratigraft area at 3 months required autografting. Another way of stating that is 96% of the area where stratigraft was placed healed and did not require a skin graft. And of course, one would expect that to be similar to the control. 83% of the burn wounds treated with stratigraft tissue alone achieved durable wound closure at 3 months, which compares favorably to 86%. So realize that even when you do a skin graft, it's not necessarily 100% successful, and you could either have a little bit of failure that then requires a long time to heal if it's small, or if it's a large enough area of failure, you might have to do a second skin graft. So doing the stratigraft compared very favorably to doing autograft. Again, as in most skin grafts, itching was the most common complaint, and there were no other serious life-threatening consequences from the use of the product. And again, no infections. That's a different bullet point. So those are things that we are currently using. What does the future hold? I think that we need to continue working on this idea of engineered skin. It's been 4 decades, but we need to continue to work on it. I think that mesenchymal stem cells and adipose tissue-derived stem cells are garnering a lot of interest, and there are a lot of proposals on how these two stem cell technologies can impact burn wound healing, either as a solution or placed within a dermal carrier and grafted. And then lastly, I think 3D bioprinting is going to continue to be looked at. as a means to achieve scarless healing. This was mentioned in the beginning, and what I hope you saw from some of the things that were presented, it takes a long time for these technologies to become available for us to be able to use, and a lot of money. So even after, for example, StrataGraft was quote unquote discovered or developed, it's requiring upwards of $250 million to take that all the way through to where we can use it in the operating room on patients. So there's a complex interplay, again, between the basic scientists, figuring out the mechanisms and how we can get these technologies, then going through all the hurdles of how it will be at the bedside, all the regulatory things that have to go through. And so this is a complex interplay that takes a long time to see something from initial discovery all the way to impacting patients' lives. And with that, I really do want to thank the Stem Cell Center. Our participation, we could not have participated in that StrataGraft study without the support of the Stem Cell Center. As busy clinicians, we did not have the infrastructure in and of ourselves to conduct a study such as that at the same time doing all our clinical duties. They really came alongside of us and helped us carry out this study. We were successful. We enrolled, I think, tied for number 3 in the number of enrollees into the study. And we are going to be participating in the next phase of that study, which is an expanded access study. And that's largely because of the collaboration of the staff of the Stem Cell Center. So I'm very thankful for it. I'm grateful for being at UC Irvine, where we have so much talent, so much energy, so much innovation, and so much passion to be able to see things from discovery all the way to the bedside. Thank you very much.
So I just really want to thank Dr. Joe and Dr. Plikus for really giving that full perspective from bench to bedside, which was part of our emphasis at the beginning. And I'm sure, although it's a bit warm in here, that many of you may have questions. So if you guys wouldn't mind joining me here, I will drag around a microphone and help you answer some for a little bit. So.
My grandmother was burned over most all of her body when she was in her 50s. And she was covered in keloid scars after all the surgeries. But my question is about her hand. Her bones and her fingers all melted. They were all in an S shape and stayed that way. I've never heard of bones melting from fire, from burns. Does that sound like something you've heard of before? I'm sure you've...
Some burn injuries are so severe that it does impact the tendon and the bones.
Her tendons were affected for sure.
So a lot of times the deformities come not because necessarily the bones were destroyed, but it could be because of the lack of good tendon structure and or the effects of the tension from scarring of the skin. It can actually start deforming tendon and bone.
For scarring, would you say that scarring is like the body's attempt at quickly healing the wound? Like a fast reacting, I guess?
Right, yes. So that would be the exact answer. That scarring, it's quick to form a scar tissue. So undergoing complex developmental processes, so the morphogenesis, it's usually very lengthy. So scar is much faster. But you know, the payback is that you do not regenerate lost functions. So, you know, for instance, hair, although it's important to us, it's not vital. But let's think of, for example, like an Arctic fox. So if two Arctic animals like fought and they have big giant wounds and they just scarred them and it's minus 50 Celsius outside, these animals will die from heat loss even if they just have a small scar on their body. So they need that fur, so they need those hair follicles. So for them, it could be honestly fatal just because of that.
Towards the end of your talk, I saw the term stem cell. I was waiting for that. I thought that over the years, I've attended seminars where they've used stem cells, isolated stem cells to generate skin. Where has that research gone? Do you know?
I've only been at UC Irvine for about 6.5 years now. Before that, I was not in academia. So largely my practice was clinical. As I've gotten into the academia and now have a chance to see more of the research proposals that are coming through, the use of stem cells in terms of generating skin, true stem cells, is still fairly immature. And one study that was seeking to use adipose-derived stem cells couldn't even recruit patients and had to be stopped. So there's been a gap in trying to figure out how to deliver the stem cells in a way that you're gonna be able to just grow skin. And so a lot of people are looking at how our current skin substitutes, which some of them are allogeneic, made from bovine collagen, how those can be used as carriers for the stem cells and then help the body then, once you lay it down, be able to get the signals and then generate skin from those. So those are active proposals that are still fairly immature. And this is speaking as someone who's reviewing some of the grants for it. They're still a little bit early and immature to garner the funding that's necessary to take them to the next level. So you're right. Most of these are regenerative techniques where we're taking cells that are further down the differentiated pathway, mostly keratinocytes, and not the true stem cell technology. I think we will get there. It just needs a little bit more time.
If a mouse with a large wound regenerates hair follicles and fat cells, why don't people?
Okay, great question again. So, yeah, there's a lot of interspecies differences. What I showed you, and I didn't mention, is the first time this ability to spontaneously regenerate haired skin was discovered in rabbits in 1948. And there was a series of studies, including one study on human from like a really prominent dermatologist at the University of Pennsylvania, to suggest that there is primitive ability to regenerate some hair follicles, certainly not to an extent that would provide a desirable, hairy skin. So there's a built-in innate ability to regenerate hairs in humans. Typically, this doesn't happen, but there is published evidence that it's possible under certain circumstances. There are some animals, for example, there are rodents called spiny mice. They're like exotic pets common in Europe. Their skin is very fragile. So if you handle them and you grab them too harsh, and they will wiggle away and the skin will rupture. And you will end up with a piece of skin in your hand and the animal will run away with a big giant wound on their back. They're kind of known for that. It's an example of what we call autotomy. So by design, loss of body parts to evade predation. So similar to what lizards can lose their tail. So these mice have an enhanced ability to regenerate. So they can regenerate much larger wounds to a much more complete extent. So now they are actually one of the most favorite model systems for studying this type of regeneration. So not laboratory mice, but what's called spiny mice. They're endemic to Northern Africa. So yeah.
So, yeah. Maybe, Max, you want to comment just a little, right? So one thing about understanding the basic science, the cues that drive stem cells, is that might be another way in terms of our future for regenerative medicine, right? If you can harness that and think about developing a therapy that's a small molecule or a driver that can stimulate regeneration when people normally wouldn't, right? So that's kind of the moonshot long-term goal from a regenerative medicine stem cell activation point of view.
Yes, so I think one way to look at it is that, well, adult tissues do not have progenitor cells, do not have stem cells. So you need to reintroduce them. So that's one way of looking at things. But in certain organs that are already originally stem cell rich, such as skin, there's actually enough progenitor cells but they're kind of clueless. So they do things in response to inflammatory signals, so the immune system generated signals, and they typically non-regenerative behaviors, typically scarring behavior. So you need to provide them with embryonic-like signals. You need to imitate signaling that's typically present in an embryo. Then the stem cells can happily participate in an alternative behavior, such as regeneration of hair, fat, and so on. So it's really about providing right signaling context. It's kind of like with kids. There's a lot of smart kids. You put them out on the street, their talent will be lost. Or, if you give them a good teacher, that could steer them toward a very successful individual. So it's the same kind of with endogenous stem cells in the skin. So.
I'm probably dating myself, but when I was a plastic surgery resident in Phoenix, before you were probably born, the big breakthrough was the use of pigskin to temporarily cover open burn wounds, which I'm almost positive isn't necessarily used today very much. But the question is, we were one of the first burn units to utilize this because the company that made the skin was in Arizona. And so my question is about the stratoderm. This is a commercially available product right now, correct? Stratigraft.
They're hoping for FDA approval, I think, by next year. So this was just completed, was a phase 3 study. So the FDA, it has to get published and the FDA has to look at it.
Okay. Is there going to be a cost issue related to this? I mean, if this is a company that's spent a lot of money developing this, is it something that's going to be an issue for treatment of burn patients or not?
That it's interesting you asked that, because that's a very good question. I was asked that question in Australia recently because I was doing a talk on disaster, and all these products are being funded through a funding mechanism that's meant to bring products down the pipeline for disaster. And so then it brings on all those questions. I mean, I would be hard pressed to say what the price point was going to be. I think for a lot of things that we use currently in burn surgery, they're expensive, and that is very true. So I don't know, and I don't know how it's going to limit its utility going forward. But back to the pigskin, there is a pigskin product that is still used, porcine dermis. It doesn't have the epidermis, and it's used as a temporary cover still in some certain circumstances.
Just a question around burn therapy. I'm not sure if all the slides were just about like fire burns or chemical burns. Is the approach different for a fire burn versus a chemical burn? And is the research approach just different also for those?
Mostly what is different between the etiology is the amount of damage that can occur. So, chemical burns, depending on contact time, can go deeper. That's why even a hot scald, right? Simple mechanism, it's just a scald. If you're burned with chicken noodle, it may behave differently if you get burned with clam chowder, just because the thicker, creamier soup is going to have a longer contact time with the skin, and for the same temperature may yield a burn that's different. Hot oil is different than hot water. The one that is very different in terms of etiology and has very significant consequences relative to the look is electrical burns. For the amount of tissue damage that an electrical burn causes, even for people who don't have a lot of visible problems, electrical burns can result in severe psychologic problems. There's higher rates of PTSD, for example, and other symptomatologies. And it's very disturbing for these patients because when they present to people who don't understand that that's a sequela, they're usually viewed as being people who are malingering. Or it's like, well, you don't even have a scar. What's the problem? But yet they have these symptoms. Some of them are neurologic, and some of them are psychologic. So electrical injury is a sort of a category unto itself because of those complications that happen from it.
So how do you factor in nutrition into your burns? Do you factor in the gut microbiome and how you can start integrating that second brain into some of your dermis regeneration?
So nutrition is vital. Not almost every patient with what we call a major burn, and many who don't even or have a moderate sized burn who cannot increase their caloric intake on their own get an enteral feeding tube, and we provide them with enteral nutrition. Without it, you're right, they will not heal, and you also risk some of the burns progressing from what should have healed and then converting to full thickness injury. So we normally, right from the beginning, when they hit the door, one of the early things that happens is they get a feeding tube and start nutrition early. What's interesting is I know there's some really interesting work on, so a lot of what happens in an ICU is the PTSD, the delirium, and things that happen from all the medications these patients are under. And it's interesting, the whole circadian rhythm and the fact that we are feeding these patients 24/7, which is very abnormal. And the question is, does that contribute to some of the delirium? I know some really nice work being done down on the UCI main campus. I was able to go to one of those collaborative meetings and started getting this thought that perhaps we should really think about how we feed patients and try to take into account that whole circadian clock that's in the gut itself too. I haven't wrapped around how to study it, but listening to that talk really brought that to light. It is very important.
So do you consider any intermittent fasting to try and cut that window down to allow for it?
I have talked about it, just kind of pontificating on rounds. Like, I'd really like to kind of change what we do, but I haven't yet made that move because it would be a huge paradigm shift for us. It would be a very large paradigm shift because we're also trying to get them to eat. So if I switched it to make it more feeding and during the day and mimic the natural cycle and not feed during the night, I already know all the things I'm going to hear. Well, then they're not going to eat and they're just going to get fed and we're not going to be able to give them enough. We have to feed them. And so if I can overcome those theoretic hurdles, I think it's a worthwhile thing to look at because delirium is getting so much attention right now in the ICU. The fact that we might be exacerbating it by how we're feeding patients would be a really interesting and important thing to look at.
Hi, I was wondering, so earlier when you were talking about skin grafting, I noticed that you guys talked a lot about thermal burns, but when it comes to, for example, if the patient has suffered cold burns or electrical burns, is the process of skin grafting different?
So we don't get much frostbite, but places that do, a lot of times the frostbite is mostly, dealing with the hands and the feet. But so I can't speak too much. I know some therapies are looking at salvage and really for frostbite, because it's looking at digits, we let them declare over a longer period of time and we try not to do things surgical in the beginning. In terms of, again, electrical, the things that we have to be concerned about with electrical burn is that the injury is a lot deeper than the surface looks at. So if you decide to just take them in a single stage of surgery, meaning, oh, I see a 3rd-degree burn, I'm going to take them, excise it, and stick a skin graft, many times those grafts will fail because the underlying tissue hasn't had a chance to, quote-unquote, declare or delineate itself to actually being damaged tissue. So you have to be concerned, and so we look into the type of electrical energy, the volts, the contact time, and we have to be concerned. And so a lot of those patients will get 2-staged, meaning we will do our initial excision and then put a skin substitute like cadaver skin and wait to let that time to delineate so that we don't have excess loss by putting skin onto a bed that eventually doesn't survive.
So maybe one question after this. We'll start wrapping up.
Hello. So I was wondering also like the repercussions like after surgery, like when you get home and like the nutrition and like the other therapies and such. Are there any other hurdles in like the aftercare the patient needs to follow, or like are there any like risks, like maybe not only like the skin needs to be grafted again, but are there other health risks that also may be affected because of the surgery?
That is a question that we could answer for the next hour.
Oh.
And it's a really good question, and it is something that when we talk about what has improved in burn care with the specialized burn teams, it's that very fact. Those of us who do burn, a lot of us fell into it, meaning I wasn't in high school saying, I think I'm going to be a burn surgeon. But the other thing that drew us is the ability that we follow patients. I'm following a patient right now that I first took care of when she was 2 years old, and she's 12 now. And they followed me from my last place and kind of came right down to UC Irvine because of that longitudinal aspect. Yes, they face many things over the years. Some of them are physical, like you said, dealing with the scarring, the contraction of the skin over time. It could be psychosocial, like I mentioned, how they reintegrate back into school, into work, with the stereotypes and the values that we have regarding skin and beauty, scarring, etc. In the immediate time frame, a lot of what they deal with is the biggest complaints from burn survivors are the pain and itch and scar. And to a varying extent, they all deal with those kind of things. And especially in that first year after burn injury.
So recently we saw like a face transplant for the first African American. So the problem with face transplant and like other cadaver-based like donations, it takes a long waiting list. So what do you feel like, are there any like comp science, computer science solutions to these problems?
So that was, That gentleman is an amazing man. We had the privilege of taking care of him during his acute injury. He is honestly one of the sickest people that I've ever taken care of from a burn injury. I did not think he would survive. He has the greatest disposition and the most positive attitude I think I've met. He considers what he went through a blessing, and he wants to be a blessing to others. That's just the kind of man he is. So I'm really happy that he was able to fulfill this journey, and just the graciousness of him and his family and the donor family for wanting to make this something that they bring attention to the issues of donation, especially among minorities. But to your point, I think the real issue is what we're talking about here is preventing it in the first place. If we can get full thickness burn injuries to heal without having to do skin grafts and get them to heal in their most natural form, just like embryonic healing, without scars and with full function, with all the appendages. So you think about how complex that task is, not just getting a covering to grow with collagen and epithelial cells, but with all the skin structures that give it its function. So you talk about what they deal with, right? So if you've got a full thickness that was just, how do you regulate your temperature? They have a really hard time. They can't sweat. When it's cold, they're freezing. When it's hot, they can't sweat enough, and so it's miserable. They're itching. They don't have the temperature regulation. It's a big deal. And so how do we obviate the need for all that? It's this research to try to produce scarless healing.
So with that, if you would just join me in thanking our two speakers for this evening. It was really great.